Good afternoon, everyone. So welcome to the next in our Andlinger Center highlight seminars for the spring. Before we get started, just a minor logistical note. This is going to sound ridiculous, but please turn off your cell phones because we were having feedback, which we've determined is due to that. In fact, you need to tell me, or David, tell me to turn off my cell phone when I sit down. For those of you that don't know me, I'm the founding director of the Andlinger Center. I'm Emily Carter, and I'm glad to see you all here to hear what I think will be a very exciting talk about leading us to hopefully the promised land of non-petroleum based fuels. From David Barry. And I just want to also take a moment to advertise that in a couple of weeks we'll have next of these Andlinger Center highlight seminars that will be on wind energy by Pratima Rangaranjan, who leads the global scientific R&D program of Vestas, which is a leading wind energy technology firm. So with that, I'm going to let Professor Jamie Link from Chemical and Biological Engineering introduce our speaker today.
All right, thanks, Emily. It's really exciting to have David here to tell us about some groundbreaking work on biofuels and making them renewably. So I'll do a very brief introduction because if I told you everything David's done, I would eat way too much into his time. So David actually did his undergrad work at MIT and also completed a PhD there in 2005. That wasn't enough, so he completed an MD at Harvard Med School in 2006. Before he even finished his MD, he joined Flagship Ventures, where he still is today. He's currently a partner, and he's been involved in the founding of several very influential companies, including LS9, which is involved in biofuels, Joule Unlimited, that we'll hear about today, I think. And in the cancer therapeutic space, he's been involved in the founding of Theracrine, and his interests extend even into things like specialty nutrients, and has recently founded a company called Essentient. So he's really been very active in trying to bring these new renewable technologies to the forefront. And so I don't eat into his time anymore. Welcome, David.
First, obviously, thank you for the opportunity to speak here today, and thanks, of course, for the kind introduction and the invitation. I'm going to speak a bit about forming companies, some of the companies I've been involved in. Originally, I was supposed to speak about one, but I have a problem of focusing for an hour on a single topic, so I have at least 3 companies in here, and I may have snuck a couple more in along the way. If my slides change along the way, bear with me. I apologize. So the title is going to be a little bit misleading because I'm not just going to talk about that. But I wanted to start out and give you a sense of what I do, where I come from. So I work at a firm called Flagship Ventures, which unfortunately makes me by title a venture capitalist, a term which I hate, and I, as a result, don't respect myself because of that. We do things that venture capitalists do, i.e. we have money and we invest in technologies, and we hope to take those investments and return more to our investors in turn so that they continue to give us more money. And that's kind of the traditional theory in venture capital, although of course of late it hasn't worked for many. But we also do something that's a little unique, where we have a group called Venture Labs. And Venture Labs, in one way, harkens back to the early days of venture capital, but in another way, when you look at it today, it's somewhat unique in the industry. And what we do in Venture Labs is really look out into the space and look for, if you will, opportunity spaces where there's the potential for technology solution, a theoretical intersection with market pull, and thus some timing by which we can actually make something that can hopefully have an impact on the world and also be able to be monetized in a way where our investors will continue to want to give us money. We do this across life sciences broadly. We do this across the field of sustainability broadly. We don't exclude any field. We've done this in IT. although not nearly as much for a whole set of institutional reasons. And I've personally been involved in about 7 companies that we've founded out of this in the last 6 or so years. The concept here is we really want to do big things. We want to be, if you will, disruptive by design. We want to figure out what industries are susceptible for disruption and how we can actually systematically do that. The approach we have uses a lot of the traditional buzzwords of entrepreneurship and venture capital, so I don't want to bore you by saying iterate over and over and over again, because you can go on YouTube and find a bunch of entertaining videos about that. But it's really about pushing innovation to the limit. And the way I describe what we do is we come up with an idea. And the idea is probably wrong, or at least it's not right. So we'll go around, we'll find a bunch of people who know something about the idea. We'll disguise kind of what we're doing and ask people a bunch of questions. And what they'll tell us, because anytime you talk to someone about a new idea, they'll tell you you're wrong and you're stupid. So we go and we hear, you're wrong, you're stupid, and this is why it's going to fail. And you go around to 50 people, you learn that you're wrong a lot and you're really, really stupid. But then you get about 100 reasons why it's going to fail. And you go back and you look at your idea and you think very critically about it. You push very hard on it. And then you go back to the same people and you ask them similar sorts of questions. And you find that you're still wrong and you're still stupid. But then they don't know why you're wrong and stupid. And when they don't know why, then you might actually be onto something. And that's when we say, well, let's put it into the lab. Let's actually put it into a wet lab and do that experiment that's going to kill it. And the reason I say that is entrepreneurs never like to do that experiment. If you think what an entrepreneur is, they're someone who starts a company, and their job is to exist in the context of that company and make that company successful. So if they ask the question that can kill the company, and the answer turns out that you're going to kill the company, they're unemployed. And it turns out people don't like to be unemployed. So it turns out most entrepreneurs don't ask the question. Some people are successful despite that, but a lot of companies will be 20, 50, $100 million in and still actually never have had the potential to work, but they've been able to handle enough people to get some money and hopefully find something that works, but they never pressure tested it enough and it doesn't actually work. So it's an important thing because we want to, everyone talks about failing early and failing fast. We don't like to fail, but we like to figure out why something might not work and figure that out early. So when I talk to less intelligent audiences, I have my picturesque way of saying we try to basically find the intersection of market pull and potential technology solution. I won't bore you with that. We look at it, as I was saying, as basically sort of an iterative cycle. I'm using that word again, I apologize. But the idea is almost we're resisting starting a company for literally as long as we can. The incubation time, although I hate that word too, has been as short as probably 3 months, but has been in certain cases upwards of 4 years, where we're going through these same experimental bases of trying to figure something out. I'm actually launching a company formally next week that has been 4 years in the making, and we're now convinced that we actually have something that's going to be pretty interesting and pretty exciting. I'm not going to talk about that today. That's not meant to be a jerk about it, but it's just a little off topic. But what we're basically doing is when we take these ideas, we put them in a proto company, and when you think about it, the people who we have working with us are all entrepreneurs, but we all suffer from massive ADD. And the second that you decide you want to actually invest in a company and put yourself into a company, that's a year, 2 years, 3 years that you can't do something else. And when you remember that, you convince yourself that this company has to be really, really special for me to want to dedicate myself and give up the things I don't yet know about for the next couple of years to make this work. And when you put that as a hurdle, then you start saying, okay, the companies that I'm going to start may actually be pretty special. But despite the fact that we put a lot of process in this and we spend a lot of time on this, we try to be very efficient. It's not useful for us to sit idly in a lab, although or in our offices, which, while we have a nice view, apparently, people don't last too long if they aren't productive, and our investors, again, don't like that. So we found 2 to 3 companies a year. We've been increasing that rate and actually looking to systematize it. We've done about 25 since about the year of 2000. I'm going to change gears a little bit and move into one of our spaces. We started thinking about sustainability, renewable fuels and chemicals, energy, water, et cetera, in about 2005. Every venture firm in the world likes to tell you that they were doing it back in 1672 because if you were doing it for a while, you must be smart. We actually started in 2005. But when you look at the space, it's kind of an entertaining space. And I actually pulled this yesterday out of a report that came from JP Morgan, where they were asking the question, this is something that they were sending out to their clients, who apparently they have a lot of clients who've been asking whether they should put their personal wealth into renewable fuels. And they actually put out the question of, should you actually bother with renewables? And they pointed out that there's an incredibly large uphill battle because you have a bunch of incumbents. You have coal, natural gas, I don't need to tell you, that control most of the market. And unfortunately in this space, it's been a market where few people survive, few ideas, few technologies survive. And when you look at the history, it's usually been a dominant technology at any given point that kind of forces out all of the others. And they pointed out 3 issues that have been crippling the field broadly, which I paraphrased as why bother, why now, and another way is why ever. The why bother is there's plenty of fossil fuels. The why now, well, only nuclear really has had a long history of a massive R&D budget, and that hasn't gotten as far as I think some people like, or in certain cases, a lot further than other people would like. And a lot of the approaches just don't make sense from an economical and thermodynamic sense. But what's interesting is there's a lot more to what's behind the verbiage. When I start looking at sort of the questions that people ask and the way people think, and I pulled this from the same report, I just thought these were entertaining, I won't take you through it in detail. It points out how limited the worldview is that a lot of people have about energy. And what they like to talk about are things that people have talked about for 30 years. The amount of innovation that's been in this space has been I think, amazingly small. So the top left up here shows actually a fact that I found pretty interesting, that the intensity, the amount of oil that's needed to generate a certain amount of GDP has been declining. People have been using that as a basis to pretend that we're not running out of oil. Anyone who's in the oil field actually recognizes that the supplies are becoming harder and harder to come by. They're becoming more expensive. So a lot of things like this come around and they suggest that, well, of course the massive electrification of the fleet, because we all drive electric cars, has made our dependency on oil decrease, so let's not focus on this anymore. And then people looking at things like, if you took all of our corn and converted it into ethanol, how much gasoline would it displace? And the answer is 8%. The problem is right now, by the way, we're using 40-50% of our corn land, and we've displaced a little bit north of 8%. So something's wrong in this math, and it just means that there's a lot of prejudice against the field. But that being said, that's just kind of a nice little set of walls that when you want to get into sustainability and renewable fuels and chemicals, you got to think about, and how you're going to convince people. So I'll tell you a little bit about my story and where we've been and some of the companies we've been playing around in the space. So I tend to be a very simple person. I like really simple rules, because in at least some form of simplicity comes at least a logical way you can think about it. So when you think about oil, ultimately over time, or at least this is the theory that people tell us, is that you had sunlight and CO2, and it made some sort of living organism, whether it was a tree or an algae, and then something magic happened and it got buried below the ground, and something then magic happened with pressure and temperature and other things that at least I don't understand. And you got oil, and then some guy, in the last 10 to 50 years ago, poked a hole in the ground and made a lot of money out of that process. And when you look at sort of the renewable space, we're basically looking at the same thing but trying to shortcut it. And so we have sunlight and CO2, and it goes into a field and it's captured by corn or switchgrass or whatever other feedstock people want to think about today. And then we pull out sugar. Keep in mind, plants are evolved to resist degradation because they live in a harsh environment, and if they got degraded regularly, they wouldn't exist, they wouldn't survive. They don't want their energy, sugar, to be digested and eaten on a regular basis, so they resist that. That's the cellulose problem, if you will. And then we want to take that thing that's really hard to get, and that's why partly costs have gone up, but other reasons too, and we want to convert that into something useful. I know I have a picture of oil here, which is useful, although people are making it into ethanol, which is good to drink. So we've done a lot of stuff in this space. I'm not going to bore you. I don't want this to sound like a pitch for the stuff that we've done, but we've looked at this as sort of a simple way to look at how do you actually streamline this process. The companies I will talk about today, I'm going to talk about LS9, I'm going to talk about Joule, and I'll talk about one other, which is actually in the nutrition space. But simply put, The innovations here, we're thinking about how do we speed up this process for Mascoma and LS9. Midori is thinking about a way of how do we more efficiently liberate sugars. And Joule and Novomer are in very different ways looking about how do we shortcut the whole process. Of course, a simple rule is the fewer steps, the more efficient. It doesn't always work that way, but it's a nice way to think about it. So I'll tell you about LS9. LS9 is a company that I founded in 2005. We had invested in Mascoma at that time. Mascoma is a cellulosic ethanol company. That's about it. At the time, in 2005, cellulosic ethanol was going through an inflection point where basically the amount that was being used and demanded by the market was increasing almost exponentially because of a government-mandated MTBE replacement curve. And most of the people, particularly venture capitalists who are getting into it, were telling you, this is a path by which we're going to go to E85. We're going to replace all our fuel with a less energy-dense thing that we don't have enough resources to make, and it's too expensive. So beyond that, it sounded great. But what was behind that, and this was actually pretty informative to me, was when you actually understand what was going on, you had some very powerful members of the corn lobby, one of whom is currently the Secretary of Agriculture, one who's a senator from Iowa, Vilcek and Grassley respectively, who were very influential in basically creating a market for corn for all intents and purposes, a subsidy for farmers. Would they admit that? Well, yes, I've had that conversation blatantly with them, and they will admit that that's exactly why ethanol came around. That's why it continues to persist, despite a series of resistance and negative LCA analyses and things along those lines. But what it pointed to us is, well, as long as you can upmarket, as long as you can upvalue sugar in some form, farmers will support that. So we asked the question of, is there something better you can make out of sugar? And basically, a lot of these answers are obvious in hindsight, but we said, well, what's a better fuel molecule than ethanol? Now, you might tell me obviously the answer is petroleum. Unfortunately, I'm a little thick-headed, so I had to put together a 300-page PowerPoint and look at every single thing I could imagine making out of sugar and building the market opportunity, the feasibility, the risks, and all of that. And, well, after all of that, I came up with the answer of it's diesel and petrochemicals, so maybe you guys could have saved me a lot of work. But it's not simple enough to think it's just petrochemical because they're energy dense. What's beautiful about them as well is oil and water don't mix, so you can imagine that your separations get easier. You also need to believe that you can do it efficiently, because if you're making energy, you need to preserve the energy in the process. And the ethanol guys were going around talking about how mass efficient going from sugar to ethanol is. It's irrelevant. It's horribly energy inefficient. So we needed to convince ourselves we could do that. We also needed to convince ourselves we could use any feedstock, but we kind of arrogantly assumed that we could at the time for the sake of argument. So what did we do? We took a look at a cellular wiring diagram, realized that we don't understand that. We made it simpler and asked, can we look at central metabolism and find any which way we can go from any compound in here to something that looks like a fuel molecule, anything that looks like diesel or a diesel-related compound, a petrochemical. And we came up with about 12. We developed our own mathematical modeling approach, ran them for energy balance, and it's not that surprising that the fatty acid biosynthesis pathway came out on top. It has about 90% 90 to 94% energy efficiency, depending on a couple of tweaks you do. The next most efficient pathway from an energy basis is the isoprenoid pathway, which is about 60%. And it's kind of obvious that it's fatty acid because when you think about how good fats are at storing energy and how hard it is to lose fat when you try to diet, well, although that's not linear, it makes sense. Fats are meant to store energy. And oh, by the way, the chemistry of fats looks a lot like diesel with a little bit of a carboxylic acid group at the end. And so we said, well, this looks pretty interesting. We patented a lot of that stuff and then wanted to go and see if it was possible. So we put $500,000 in and tried to see if we could actually find the genes to make this work. Now, over a relatively short period of time, we first found genes that would allow us to go make alcohols, then we found genes that would allow us to make esters, then olefins, and only a while later did we actually find things that can make alkanes and branched alkanes and things along those lines. A lot of these genes exist in nature. They don't exist together, unfortunately, so we had to do a lot of genetic mining, put this all together, and what it gave us was a chassis. We use coli and other such organisms by which we could use fatty acid biosynthesis use some of the traditional art to massively increase the productivity and the flux of sugar through the fatty acid biosynthesis pathway by deleting some of the other sorts of things. And then when we put the right genes into the organism, we can get the right product out. I wish it were just that easy. The other thing you have to do is make sure you're doing it in a highly appropriate way and a highly efficient way. The cell is really good at telling you when it doesn't like things because it dies. And I thought you made a great comment beforehand that these cells are not very productive when they're dead. So they need to be alive. And when you're making things from the fatty acid biosynthesis pathway, fatty acid biosynthesis uses a lot of NADPH. So you need to figure out ways that you can shortcut sugar biology to give you excess supplies of NADPH. The good thing is nature's done that. We've been able to do that, and that's allowed our efficiencies to go up. The other element of this is, as I mentioned, we liked fats because we thought oil and water would separate. We made that as an assumption, and one of the things I always like to remind people is you got to test your assumption. We started to outsmart ourselves in a way because when you have water, and then you have an organism which is basically a sack of water with some fat around it, and then you have some fat. Is it going to separate, or are you going to form some really ugly micelle mixture, or what's going to happen? And it was funny because the guys who were in the lab, I was not one of them, didn't do that experiment for about a year. They didn't think about it. And I actually suggested one day, I said, why don't you just take some cells, they don't have to express something, put some oil, put some water and some cells together and see what happens. Well, the good thing is it turns out they separated. It was just kind of an interesting lesson of how sometimes people don't ask these questions and they get themselves caught up in these conundrums. And you get these nice pools that come out. This is a nice obvious cartoon drawing where literally if you take it out of a fermenter, you separate it, you let it sit for after about a minute and a half, you'll get a pellicle that starts to form on the surface and you can almost scrape the oil off. It's very easy to separate. When you put that together, you get a very efficient process by which you can make hydrocarbons. So literally we go from feedstock through our fermentation process, and that's our final product. I'm not going to talk about this in a competitive way. Every other process that's out there, if you want to get to diesel, has a whole series of other steps that they have to make. And our idea is, let's take these are mass efficiencies. Let's take the highest energy efficiency and the simplest process and get to diesel, and we'll be able to do it at a cost point that matters. Because at the end of the day, the fuel and chemical markets turn out to have intelligent people in there that are not going to pay more just because it's green and pretty and grown in a steel tank. And they're not going to pay for a worse product. They want the same product at a equivalent or lower price. And so what we can do is we can deliver drop-in replacement diesel at an equivalent price of about $60 a barrel. What's even better is the fatty alcohols, for example, which price at about or sell at about 5 times the cost of crude because of some interesting supply-demand dynamics. We can also make at $60 a barrel, but they trade right now at $500 a barrel. So we get the opportunity to very quickly get to market, but also have larger margins, because I'm not going to sell it to them for my cost of goods. I'm going to sell it to them to make some money as well.
So when you say that you've made ultra-clean diesel,
Yep.
By that do you mean biodiesel, like what you get from soybeans and things like that?
No.
Or do you mean, so can you say something about the molecular mixture of the diesel? that you're
Sure.
talking about?
So soybean diesel is an alkyl ester. Our diesel is a straight-chain hydrocarbon.
Okay.
So if you tell us what you want, if you want
So it has no oxygen in it?
no oxygen.
And is it unsaturated?
It can be. We make it that way. If people want saturations, we can do that too. There's reasons you want both. So we just tailor basically on the need of the potential customer. And the good thing is customers are very picky. So we just ask them what they want. They tend to tell us, and we make it, and then they tell us it was actually something else. They were just testing us. And then we go and make that.
You can control the chain length?
Yes. That's actually part of the beauty of the fatty acid biosynthesis pathway is it's an iterative loop. I'm using that word again, but here I guess I mean it. And there is a set of enzymes that you can use very precisely to cut off that loop at any given point. And the good thing is you add it pairwise, so you can go 2, 4, 6, 8. We don't do things in the 2 to 4 range for a set of reasons, but we'll do anything we've done things between 5 and 30. And there's some magic to do 5 to 8 versus 8 to 14 versus 14 to 20 versus 20 to 30. It's a slightly different way of doing it, and there's slightly different genes that are necessary. Yes, yes. So, yeah.
What are you feeding the cells?
Sure. We're literally we use a standard nutrient broth, and right now we're just using industrial grade sugar. We're not doing anything fancy. We've engineered the cells so you can use 5 carbon sugars and 6 carbon sugars as well. We actually have a science paper on that, because I think it's the first time that cola has been engineered to do that, as far as I know. I could be wrong. So where we are now, this was an idea in 2005. It's now raised about $90 million. We've gone through a pilot plant. We ran a series of demonstration plants for a number of years, and later this year we'll open an 135,000 liter plant in Okeechobee, Florida. I'm going to switch gears for a second, or this is more of an evolution. So going back to my original chart, the problem with this approach is you use sugar. And sugar, it turns out, is not the best feedstock because it's expensive. And the more LS9 and its colleagues and competitors are successful, the more Brazil is going to make a lot of money, or at least that's what people surmise. And so we asked the question of, can we eliminate the sugar feedstock? And as you think about that, and as you think about transitioning into using sunlight and CO2, you think about the other issues that come along with sugar. So we just put a bunch of questions out there of, can we make a drop-in replacement fuel at a cost-competitive price that actually beats what, say, Exxon, BP, and others are thinking about for the price of the next barrel of oil, but do it so it's independent of arable land, independent of fresh water, and with a wonderful greenhouse gas profile. We could put some other requirements on there if we wanted to, but we thought that was good enough. Our approach was basically say, let's, for all intents and purposes, take the same approach that nature has, sunlight and CO2, not the randomness and the unknown portion, and go directly to that end product in as simple as a process as we can. Again, here our approach is to make diesel and other drop-in replacements. We make pure compounds, not mixes, and that's important for a whole set of reasons. But it plays very well into what the refineries want, so they can upgrade things. None of these companies is going to replace an entire industry, at least in the foreseeable future. And so recognizing what you can actually do in the industry is important. So what did we do here? Well, we looked at this in 2 ways. First is, we asked the question of what's good about biology, what's bad about biology. What's good about biology is it's precise. You can make biology do what you want. I've been accused in various emails of enslaving organisms, which is entertaining. So the more you can use biology for precision, great. The bad thing about biology, it scales like hell. You have to scale in 3 dimensions, and when you scale in 3 dimensions, things are unpredictable. The beauty of sunlight is, for all intents and purposes, sunlight is 2-dimensional because it hits the earth, and the amount of photons that you get in a given unit area is your amount of input that you ultimately get, and that's it. And the solar guys actually got this right because they scale in a totally modular way, but they also basically scale in 2 dimensions. So you build a solar panel, you know it works, you can go build another, and you're pretty sure it's going to work. You can go build 1 million more, and they're going to work. So you've taken out the scaling factor. So we actually asked the question of how do we harness all these benefits? So first we looked at the biology. When you look at what algae does, what trees do, what natural organisms do, is they capture sunlight, CO2, and water, and they use that to grow and maintain themselves. And although this isn't the intent, they secrete oxygen as a byproduct. That's really inefficient for us because we have no interest in the cells kind of enjoying their lives. We're not, here to kind of give them the nice beach house in Miami or something along those lines. We want them to work hard for us. And what we decided to do is try to create them into catalysts. This is what kind of biotechnology has done really well for 40, 50, 70, depending on whose estimate, 4,000 some odd years. And what is done in that space is you either take a cell and you engineer it to make something expensive that you keep inside the cell. Or if you want to lower the cost, you make it secreted. And people have done this for a while. It just hadn't been done in photosynthetic organisms. When we looked back in 2007, when we started this company, there were 2 papers about a genetic engineering of a photosynthetic organism. And so we went to do it. We went out to try to do that for the first time. But we wanted to do it in such a way where we made sure that the cell was going to take in its input, make its output, and secrete it. If it didn't do that, we wouldn't go forward. So the good thing is, as I said, people had been doing this for a while. The reason I say 4,000 years is, of course, wine is probably the first great industrial product that came out of this sort of technique, although they probably didn't know some of the stuff we do now. Maybe they knew more because they had wine and focused more on it. But what happened since the 1940s through 2007, for all intents and purposes, gave us a roadmap of the tools that we needed. Unfortunately, that was nothing more than a roadmap because the cells behave incredibly differently. We're kind of taught in genetics class that every cell has its chromosome, and the chromosome is what happens, and it divides, and then the cell splits evenly, and all of that. These cells behave quite differently. So we use cyanobacteria. A lot of cyanobacteria have a chromosome. All cyanobacteria have a chromosome. But that chromosome, not during the cell division phase, arbitrarily replicates a certain number of times. That averages about 7. It's not 7 distinct chromosomes, it's the main chromosome that's replicated 7 times. On top of that, it has 7 plasmids. And these plasmids each replicate a random number of times that ranges between 16 and 108,000. And when you think about what you do in genetic engineering, you take a foreign gene, you get into the chromosome, it divides, and both daughter cells get it. Well, if I have 108,000 copies, one of which has what I'm trying to put in, and the other 107,999 don't, my daughter cells are not both going to have the same thing. So we had to actually figure out entirely new ways of engineering these cells. And we've developed an entire art around this. By the way, there's actually reasons why these organisms do that. They sit out in the sun. The sun is a great mutagen. These things don't want to mutate. So when the sun mutates the DNA, what they have this ability to do is divide very asymmetrically. So they can take a plasmid, they can take a chromosome. Their division can be something as simple as one plasmid gets pushed off, and now the cell remains pure, it has no DNA damage anymore. And so it hates people like us who are trying to, if you will, brainwash these cells. So that was the first thing. We needed to figure out how to genetically manipulate them. After we did that, we needed to figure out how to control them. There's a lot of complexity in these cells. So we used a lot of the modern techniques. We used gene arrays, did a lot of metabolic mapping, et cetera, et cetera. This, just for whatever it's worth, represents the Calvin cycle, glycolysis, the Krebs cycle, et cetera. But it looks very different here than the stuff that I learned when I was an undergrad or in grad school. And the regulation of this is a real pain in the ass. Because what happens is these cells, and it's not surprising, have a system that's dependent on the sun that governs their entire circadian rhythm. It's proteins called Chi A, B, and C. And they have this really elegant dance where they rotate through the cell. I'm not going to bore you with all that sort of stuff, and I don't have any cool videos, so don't worry. But basically hits a whole series of all the major transcription factors that controls the cell. And we looked at that and we said, well, that's kind of bad because unless we can control this thing, we're never going to be able to control this thing. So we figured out how to completely unwind the entire regulatory mechanism. Then we looked at what all the successful people in the past have been able to do, and you need to also be able to decouple growth and production. So we developed ways to be able to do that. And now we have a system by which we can grow up cells for 3 days, turn them over to steady state, then they operate for 7.5 weeks, and take in the same inputs and secrete diesel.
So you don't get diesel until 7.5 weeks?
No.
Producing diesel throughout?
Throughout. For 7.5 weeks, we're producing diesel.
Are you going to talk about the rate of production?
That? So,
Well,
yes.
count the rates.
Well, so I wasn't going to get into the transcription factors and how all those things come together. But one of the things that's important, and I'm happy to share some of the results of this. This is actually relatively consistent with this. So one of the things that people tell me all the time is, this will never work. You're stupid. And the reason it'll never work is because photosynthesis has an efficiency of 1%. And you ask people where that comes from, and that's because most plants and most algae, when you let them on their own, have a net areal light efficiency to material capture rate. That's 1%. That does not mean that photosynthetic efficiency is 1%. So pretty consistent with what I think you're getting at. What we actually did before we started was we looked at all the things that are relative to the cell and how we could actually control those rates. And I'll come back to one element where the cycling becomes really important, one element where controlling the Chi A, B, and C factor becomes really important, and ultimately when you convert these to behave as catalysts, when you get the light-dark cycle right, and again I'll come back to that in a second, you can actually shift that efficiency, and we've done this in the lab, to move from 1% or 2% up to 7%, And the theoretical max that we're looking at right now is about 12%. This involves no engineering of the photosynthetic mechanism. This is not changing it at all. It's optimizing the way that the energy that's captured by photosynthesis is used and the way that energy is delivered to the photosynthetic machinery. Unfortunately, there's just a doctrine that exists in the photosynthetic literature that suggests photosynthesis is very inefficient. And I personally believe it's just because it hasn't been tapped. We have produced thus far, we started out making ethanol. We've produced ethanol at 15,000 gallons per acre per year thus far, which is equivalent to about a 7% photosynthetic efficiency. You can't get that. You can just do the simple thermodynamics of it. You can't get that unless you have 7% photosynthetic efficiency. Our theoretical max for ethanol is 25,000 gallons per acre per year. The theoretical max for ethanol, for diesel, is 15,000 gallons per acre per year. It's the same number, it's just that ethanol and diesel are different molecules and there's a different process that you have to go through. And these are just some comparables, because those numbers are actually pretty game-changing when one looks at it. But part of the reason I was telling you about how we engineer the cell is the single most important thing to have here is a chassis. You know, I don't care what fuel or product we make to start. If you can capture sunlight and get it into central metabolism, you can do a ton of things with it. The reason we started with ethanol is it's 2 genes. It's really simple, or we thought it was going to be really simple. But what it taught us is, first and foremost, the cofactors and the regulation of these cells is totally different. We actually had to completely rewire the way ethanol is synthesized because we had to change the cofactors from what people had done in E. coli. But after we had we changed glycolysis, changed the regulatory mechanism, changed all the cycles, started getting massive efficiencies, we had a chassis that we could drop any fuel and any chemical into. And the slower timeline that we got with ethanol gives us a chassis by which we've been able to move diesel along quite quickly as a result. And this offers us a pathway by which you could look at central metabolism, you look at the sort of common things that exist there, and almost use your imagination and say, what do I want to make? You can make ethylene, propylene, which are the 2 biggest chemical commodities that exist today, which are $180 billion in markets each. You can make fatty alcohols, fatty esters, you can make diesel, you can make olefins, you can go any which way you want. And once you have that central chassis, all of the stuff is possible with minor tweaks to your efficiency based on what the compound looks like and what the downstream ultimately is. The other elements of this, the biology is important, but it's not sufficient. You need the biology to work in a catalytic fashion. But first of all, if we're genetically modifying it, you got to enclose it, otherwise the EPA will never approve it. But once you realize you're going to enclose it, you start to realize you get all sorts of benefits from this. So when we go look at plants, not the steel things, but the green things outside, we tend to think that they like sunlight, and the more sunlight you shine on them, the better. The photosynthetic mechanism is odd in that it has an antenna that captures light. It goes in through a plastocyan and plastoquinone, et cetera, mechanism, and the energy is thus captured. But the antenna can only store a certain amount of energy. If you have excess energy on that antenna, it gets shunted off as IR. That creates heat, obviously. And cells tend to like to exist within about a 10-degree range and are productive within a 10-degree range. So the more heat we have, the more cooling we have to do, the more energy it is. So we don't want IR. What's interesting is when you look at the cycle that actually maximally gets photon utilization, thus prevents photon loss, thus improves net areal photosynthetic efficiency, you actually want these things in the light for about 0.1 seconds and in the dark for 0.9 seconds. Why nature would have evolved that, I have no idea. That's just empirically shown. And if you strobe lights at that rate, your growth rate of a cell will be 4 times higher. There's data on that, by the way, going back 30 years. This isn't just me saying that. Now, as you can probably tell by the fact that this is sitting out in a field with a blue sky, we don't have a massive strobe light that's floating over these things. But our solution to this was, well, these things are going to have to move. We don't want them sitting there in stagnant ponds. They may like that, but again, we're not here for their interests. We want a light zone and we want a dark zone. And we use CO2 that's injected in, the CO2 is off gas, and we use this to create a mixture of turbulent and slug flow that on average has a given cell in the light. We've actually changed the machinery so that it's closer to about 0.3, 0.7, that it's in the light for about 0.3 seconds, and in the dark for 0.7. We actually would like to change that even more because the more dark volume you have, the more movement you have to have, the more energy it takes, the more cost you have, et cetera, et cetera. This is a highly multivariate equation that we have to solve at the end of the day. We also play some games with the surface. You definitely can't see it because, well, I don't even think it's shown here since this is a schematic, by which we can do things like nanotexturing and things along those lines to reflect IR. We can shift wavelengths, so we can use UV, expand, if you will, the photosynthetic active region of the spectrum, et cetera, et cetera. This is our 4th generation reactor. This is actually from our pilot plant in Leander, Texas. In this case, we actually designed these reactors to look like solar panels. And we're not very imaginative, I apologize. And the idea was we just want them to be linear, scalable, simple. This is about 4 feet by 8 feet. We put 10 of them together. It's about 40 feet long. That's not accidental. That's the size of a flatbed truck so that we could ship it anywhere. This design, just so you guys know, it sucks. We never would have known unless we actually built it, but the design's horrible. And any engineer who's built something that moves fluid around would probably look at this and kind of laugh at us, and some of them did, because we have so many valves and so many interconnects and so much complexity in the system, with things bubbling up by airlift and flowing back down with no fluid, and using a header to capture our process, that it just, we got leaks, we got contamination, we had all sorts of issues. The good thing is we learned our problems, and the reason that you pilot things is to learn your issues. So we've actually developed a new Gen 5 reactor, which is actually about 100 feet long, takes a lot of the principles that worked from the last thing in terms of slug and turbulent flow, extends it over a longer period of time, uses less energy, is much more passive in the way that we manage heating and cooling. Again, leverages the linear scaling that we get. I apologize for this figure. We've contracted with Fluor, who's helping us build our commercial plant in New Mexico. The first 5 acres will be open by the middle of this year. We have about 5,000 acres in New Mexico that this is going to go on. And they gave us this picture of what it's supposed to look like. I really hope it doesn't look like this for a lot of reasons. I don't know why this truck and this car are hanging out here, but that just doesn't seem like a good use of time. And I don't know why we need trucks for this sort of thing. But ultimately, the vision that we have.
I'm missing, I lost the track here.
Yep.
What happens at night?
Very simple. There's no sun,
That was my problem.
so we shut it down.
So what do the cells think?
Well, the cells are used to living at day and night,
I understand that.
and they just stop producing.
So they are maintaining their day-night cycle, and so they're doing the metabolism goes one way in the day and the other way in the night?
So they actually go through a repair phase. This is the nice part about it. So our productivity numbers are based on NREL data that assumes cloud cover, assumes that days and nights exist, and that we have a standard installation rate. So it's not assuming that we have sunlight going
No, I
24 hours.
understand that.
Well, I'm just putting that out there. 'Cause-
I was interested
yeah.
in the biology.
But what
I trust you
happens
on the numbers.
is the cells actually, they go into sort of a sleep mode, not quiescence, but they go into a sort of a sleep mode where they actually repair their photosynthetic mechanism. Their metabolism shifts mildly. One of the things we have to pay a lot of attention to is what happens right at the change from day to night and night to day, because you have a startup time. And part of the way we actually control that is with temperature. So we let the temperature drop off at the end of the day, and that almost shocks the cells into more of this rest phase. We do get some productivity because it's
So they because during the night they make sugar, right?
not here, but ordinarily they do.
Normal plants.
Normally, yeah. We don't let them. They make diesel at night, but less.
Yeah.
And then we give them enough leeway at night to make the stuff that they need to repair their photosystems and all of that stuff. But that's the beauty of it, is we get a continuous process where we actually have time for repair.
So, do the cells need to be refreshed every 7.5 weeks, or how long can they operate without me?
So I'll answer that with a more complicated answer. So we kill the cultures every 8 weeks. We do that intentionally. We've been able to get them to go for 12 weeks. We can probably get it to go for longer. We don't want to. The reason is we want to maintain exquisite control. And with any culture, over time, you start to get little genetic drift. Your productivity goes down. We have about 95% of our original productivity at about the 8-week time frame. And so we just say, that's enough, let's kill it. It's sort of an arbitrary number. The other reason we kill it is we want to maintain control in another way, which is, it means every 8 weeks you could put a new fuel and a new chemical in there, and that gives us a lot of diversification ability. The other question that one has to ask is, what's your business that you're ultimately in? And are we going to make and own all our plants? Probably not. So this is a way where we get sort of a razor blade model of actually having a plant that gets built, and then we sell them a seed that they have to put in every 8 weeks. And if they don't purchase it every 8 weeks, they're violating our intellectual property, they're violating the agreement, we cut them off. So a little more engineering than just on the physical side.
What's the volume, if you were to relate the production volume compared to a typical, catalysis production of some fuel?
We're a lot worse on a volumetric basis.
Yeah.
We need a lot of volume to make product.
And it's slow, right?
Yeah.
That's
Yeah.
what I was worrying about.
It's not it would be a worry if separations weren't easy. It would be nearly impossible if we did an intracellular process, and that's partly why we don't. You can imagine we're covering 5,000 acres, covering a given acre, and you get 15,000 gallons per acre per year. That's a relatively slow rate. I could give you the exact number if I knew it off the top of my head. I don't. And you can imagine that it's kind of like a sheen. So you have to be a little creative in your separations process. It's not as simple as LS9s if you turn off the reactor and you get a layer up top. We actually do a little bit more of a continuous pull on the fluid and an active centrifugation. And you get small amounts of the diesel that come out on a pretty regular basis. And then we recycle actually the material to go right back into our system. So it's a relatively it's simple yet complicated at the same time to make sure that it's that we keep the amount of diesel in the process low. Cells don't like diesel; it dissolves the membrane, and so that's why we do it actively. We kind of have a good understanding where that toxic level is, and we just make sure we stay below that. But we want to do as little centrifugation as possible because that's energy and effort, and time, expense.
So you're running a giant cream separator all the time.
Basically, yeah, it's a good way of looking at it. Yeah.
So when you fill it at 8 weeks, presumably then you have to sterilize the whole
Yep.
field there with maybe bleach or something. Does that disposal of that, whatever you're using to sterilize it, does that present any kind of environmental issues?
Not yet. So we don't use bleach.
Scaled up when you had it?
Yeah, no. So you have to work with the EPA on any of this stuff. We've proposed 2 approaches to the EPA, and they like both of them. One is using ozone, and one is using E-beams. And they're not perfect. They're good for sterilization. They're not perfect from an environmental standpoint.
Does it take a lot of energy?
It doesn't take zero energy, but the amount of energy that it takes is negligible in the system. And the other thing is, it would kill us if we had 5,000 acres and you killed everything at the same time. So we do everything sort of, if you think of sort of a fed batch system, we keep everything kind of rotating through the plant as to what's going to be active, what we're killing, and what not. We may find that we want to extend the time to 12 weeks just to give us more time in between killings. It's a relatively moot difference at the end of the day. But our model right now is when the cells are spent, we've told the EPA we're going to burn them. It's not the most environmentally friendly thing, but it's the simplest thing because it gets rid of the modified genetic material. There's no questions about that, and there's a ton of opportunities of what you can do with the cells at the end of the day. At the end of the day, we got to get this practiced and working before we start getting too fancy with it. There's enough moving parts in this that if we tried to get fancy from day one, we'll kill ourselves before we get to day one. Yeah.
I understand when one of the growing algae that they have this reactor that growing up vertical. Would it be more efficient because you have a very thin reactor that will catch the sunlight?
I've never understood the logic on the vertical algae reactors. I've seen them. Photons basically hit the Earth on a given unit volume, and it's going to come in some way or another. And so any orientation you choose is going to have a fundamental limitation of photons. There's no way of cheating to get more photons. Even if you concentrate light, but using some magnification or lens or something along those lines, you're going to lose a certain amount of the energy just by actually having gone through that process. It's the unfortunate reality of it. And our attitude is, if you have to build something vertically, then you have to spend energy to keep it vertical. One of the things people tend to not think about before they build big plants is that if you're in a place like Florida where there's a lot of sunlight, there's also hurricanes. If you're in Nebraska or the Midwest, Texas, you get tornadoes. And you need things that are going to actually be able to, they don't have to withstand it, but they need to actually, you need to have a plan for when that happens, not pray to God that it doesn't happen. And I think when I look at the vertical ones that are especially inside greenhouses, the capital cost on those things is more than a solar plant. Our capital cost is about 1/5 of a solar plant. So infrastructure-wise, I won't bore you too much with this. Our intent right now is that we're working with places like power plants. We take CO2 as off-gas. Off-gas, by the way, does not mean we put a pipe on the stack. You can take the CO2 and actually powderize it. There's a number of companies that do this today. So you don't even need to be near the power plant, but there's benefits of it. You take the CO2, use it as a feedstock. We like pressures between about 1 and 4 bar plus or minus. The output that we get, the byproduct, is oxygen. Part of the reason why we're planning on burning the cells is if we're working right next to a power plant, you put the oxygen back into the burner, it burns at a higher temperature, it's more efficient. We've just made a cogen. By the way, then we can put the cells in there too, and they're burning stuff for free. So we get a lot of systems advantages that come out of this. What we're doing is eligible in some metrics for carbon credits, but our business model is not dependent on it. And we have the ability to produce diesel at prices as low as about $20 a barrel. We've done a series of third-party life cycle analyses to look at carbon. If we connect to a coal plant and we take energy from a coal plant for a heating and cooling for our heating and cooling needs, and we take the CO2, our life cycle emissions is reduced by about 50%. If we do it with a solar plant where we don't get the cogen benefits, I don't know where to put the oxygen into silicon yet, it comes down by closer to about 80 or so percent. So it's not perfect, it's not CO2 zero, but we're looking at this as an opportunity to move forward. Some of this stuff is obvious, just saying that the higher the solar intensity, the higher productivity. I'll give you a snapshot of where we are today, and I'll run through one thing just in the last 5 minutes relatively quickly. We've scaled to our pilot plant and then to our commercial plant. We've raised about $110, $120 million. We have about 110 people working. The thing that we really like about this is the opportunity where it actually can make a difference. I won't read this to you, but the point is, when you even think about what we're doing in New Mexico and what that can ultimately translate into, which will be ultimately a little less than about 7,000 barrels per day, which you could say is trivial. It's basically the new discovery rate that you get with the oil majors. And so where we look that we can start playing is actually starting to make a dent at that level. We're not going to move the entirety of America onto our oil by tomorrow, but we think we can actually start making a difference because the cost of next barrel of oil for Exxon is 55, for BP it's 62. We're producing it less, and we're producing it without the risks associated with typical exploration and discovery. In the last couple of minutes, I'll just tease you with this nutrition thing. So I got bored when I got fired, or I guess I didn't get fired. I found my replacement at Juul. That's kind of my job. I joke that I get fired, but I don't have a good severance package. In 2009, I guess I was stuck by unemployment again and got interested in nutrition. People ask me why nutrition. Well, oil and gas is the world's largest industry. You would probably assume that nutrition is probably the second largest industry, food and nutrition. It's not. Organized crime is. I don't know how to do something there yet, but food and nutrition is the third. If any of you guys have ideas on organized crime, let me know. I'm interested in. But we looked at this problem in 2009, and it was kind of an interesting challenge because we have a world. We talk about the food versus fuel problem. We look at resource constraints, and this is the problem that people don't like to talk about because we have no one actually really offers a solution right now. So. This is a quote, but when you think about it, ultimately we're facing a system where agriculture and farming today produces food that we say is enough But for the 7 billion people we have in the world, 1 million people are chronically undernourished. Another 2 billion people are regularly malnourished. We have an agriculture and farming system that's responsible for more biodiversity loss than anything else and releases more CO2 than the entire transportation sector. And we say it's not a problem? By the way, it also uses 40% of all land, not arable land, of all land, and 75% of all fresh water. We also like to think that here in America, we're not susceptible to it, but we actually don't get very good nutrition here because of, I don't want to get into the politics of it, but the nature of our nutrition system. And the leading causes of morbidity and mortality in America, obesity, cancer, diabetes, cardiovascular disease, are actually all driven by improper nutrition. And these are things actually, by the way, that people understand. So I got interested a few years ago in trying to understand this and seeing if we can do something different. So we're not looking to replace food. I have no interest in replacing food. I like food. I eat a lot of food. I'm very interested in finding the best restaurant in any given place I go. But what our food system does, and I'll talk about this in two different ways, our food system uses a very complex system to give us what we know today as food. Food has two things in it. One are the things that we need to live, and the other stuff. And the other stuff actually, in certain cases, prevents the absorption of the stuff that we need. So our approach is just make the stuff that we need. We focus on proteins. I'll come back to that in a second. The other thing, and the reason why we're never going to replace food, if you ever say to someone to change their diet or anything along those lines, they resist you. Why do they resist you? Food's social. It's part of our social fabric. It's part of who we are. We go to meals with people because it's a forum to talk to people. It goes back to even the campfire days of what do you do around campfires? You cook. Maybe that's not what Homo erectus thought about it, but that's at least the way I look at it. So we have no interest in changing food, just nutrition. So similar to everything else, I won't get into the details here. We've created a cellular factory as well with an entirely new system of how we evolve them, where we can actually produce more diversity in these organisms than you can get if you took last year's GMO rate that you would get if you'd been using that since the beginning of agriculture. The beginning of agriculture is only about 6,000 years ago, plus or minus, depending on who you believe. I don't know how we actually know that number. But if you took twice that long, you can do that. You can get that in one day in our lab. It's not just about that. We want to make the best stuff on Earth. And no, it's not in Snapple, don't worry. And what we did is we basically started this thing that we like to call the Food Genome Project. It sounds a little bit like a band name. But basically what we did is we took every food that you can imagine on the shelf. We took cows, we took chickens, we took shrimp. We didn't just take meat products. We took fruits and vegetables and made them into goo and ran them on 2D gels and characterized every protein that's in there. And we made a massive database of every single protein that exists that we could find commonly in food, in people's diets, not something that like the Incas ate on the side of a mountain at 3 p.m. to celebrate some ceremony, and developed a proprietary in silico method to measure their goodness a whole series of axes. And this has given us the ability to create a series of performance products that are off the charts. I'll give you an example. We have a product that can grow muscle better than testosterone. This has actually been seen in people. You get about 4 to 5 times the growth of muscle compared to testosterone without working out. And you get none of the side effects like anger management and body dysmorphism in the various ways that we know of. And it's actually cool. Most of us have been taught that you go to the gym to build muscle and you got to tear the muscle to build it. That's great. If you go and you do that and you don't have proper nutrition, you on average will repair the muscle. You won't build muscle. You need protein nutrition to actually build the muscle. But it turns out if you get the right dose of protein and the right protein, you can build it without the exercise, which has a lot of people kind of excited because I don't know too many people who actually like going to the gym for the purpose of going to the gym. But the other thing is, we're interested in actually transforming global nutrition here. We want to make a difference. We care about the 3 billion people who aren't getting adequate nutrition. And the system that we're looking at today is aimed at improving productivities. It turns out, as you can imagine, a cow is not the most effective means to transduce solar energy into protein. So we've developed a system that tries to shortcut a lot of those pathways and ultimately have the potential to get up to 220,000 pounds per acre per year. What does that mean? The world record in agriculture, which has been achieved once using soy on the south side of a mountain, on the south side of a hill, is 3,600 pounds per acre per year. And the average for soy, which is the most productive plant, is about 700. The implications of this are that if you wanted to provide enough protein nutrition to provide the baseline protein to all 1 billion undernourished people, it would take about 250 square miles. If you wanted to provide it for all 7 billion people in the world, it would take the land area the size of Rhode Island. It's been advocated by several, including me, that might be a better use of Rhode Island. But by the way, Senator Whitehouse from Rhode Island has also supported that concept, but that probably shouldn't leave the room. But this is something that's more close to our global mission. We're working with the Gates Foundation, with the Clinton Foundation. We have an agreement with the Prime Minister of Ethiopia on this as well. And if I have 2 minutes, I'll tell a quick story about that. One of the things, as I said, we like to pressure test our ideas very heavily. And I couldn't believe that we could transform global nutrition unless I could get, at the Prime Minister level, someone to buy in that this technique would be interesting. So through a series of connections, very early on, we got a meeting with the Prime Minister of Ethiopia during UN Week in New York. And was in his hotel room and gave him, a 20-minute pitch. And he was sitting there quietly for the whole time, not smiling, not anything. No idea how to read the guy. He has a very good poker face, apparently. And after I finished, he paused and sits there looking at me, and he said, literally puts his hands like this and says, "Interesting." And I'm sitting there like, how fast can I get out of this room? And he said, in my country, 85% of the population are farmers. And if this does what you say it's going to do, they won't have a job anymore. They won't be able to work. And now I'm saying, I see the door over there. How do I get out of here as fast as possible? And then he pauses for what felt like probably the longest pause of my life, although it was probably like 6 seconds or something like that, and says, and that's a good thing. He says, most of the farmers in my country barely even count as subsistence farmers. They barely make enough on their own to live. What I would love to do is move farming into 20% of the population, use this sort of system to give us a baseline for our nutrition, and move the other 65% into new industries that we can create and actually make Ethiopia a country that can have technologies and an economic future. And so that was a big signal for me, and it was actually quite exciting. And well, I always get the question of how do you make that happen? That's why we're working with Gates and Clinton and lots of governmental organizations, because that's not something one company does on their own. You don't shift country economies. But that's the thing that we're really excited about, is if something like this might actually work, we might be able to hopefully change the world. So that's basically what I had prepared today. I know I talked through a lot of stuff relatively quickly, but hopefully that gives a little bit of a flavor of some of the stuff we've been working on, and some of the concepts behind what we like to think about at Venture Labs, of trying to take on big problems where no one wants you to succeed and ignoring everyone telling you you're stupid.
We have time for a few more questions.
Yeah, so what we're making, we make pure powder type of products. We have proteins with proper protein nutrition that taste like sugar. We have proteins that taste like fat. You literally can't tell the difference between them. We're not making finished products. We're very specifically not making foods. We make things that go into the food system. I don't want to scare people with how the food system works, but these are things that will blend right into the standard production systems. And part of the thing that we've looked at is, just like with the oil companies, when we make oil, we're not going to go and make a gas station. We go sell to the refiners and the blenders. Well, for us, the blenders. And then they distribute it. Here we would sell it to the food processors. And companies like Pepsi and Nestle have very active interests in a lot of these countries as emerging, developing, growing markets. So we see that as collaborations. We're very actively having discussions along those lines.
Can you
Pass the microphone
to
down.
the next person?
Yeah.
So I guess first of all, with food, somebody just like a few weeks ago ate the first lab-grown hamburger, and I'm sure that requires a lot of sugar input. So I wonder if it would be interesting to take some of the outputs from what you're creating and then use that to feed whatever cells are growing this hamburger meat. And then a more important question, more relevant question, I guess, is with Joule Unlimited, your cells there require a high concentration of CO2, from what I understand. And you're siting them nearby coal plants, but does that present an upper limit on how much you will eventually be able to produce using that model? And if it does, how much does the efficiency drop if you don't have this extra CO2 that you're adding?
Yeah, so on the first question, the less important one, which is the more fun one to answer. First of all, by the way, that's not the first time this has been done. They just acted like it was. People have been doing this thing since the 70s in various ways. Less refined, and not using stem cells, which is what these guys do. The cost, by the way, for that patty was $340,000.
Yeah,
So,
that's why I was hoping you could bring it down.
yeah, I have no interest in supporting that. I have no interest in replacing food or changing food for personal, for ethical, for a whole series of reasons. And the MD side of me knows that it's a really bad idea to cause people to ingest stem cells. So I don't even want to think about that path. But on the second question, so I use coal plants as one example. We can use any CO2 source. Coal happens to be one of the worst ones to use, but that's not the point. The off-gas coal is typically between about 9 and 13% CO2. We need about 4% for it to actually work, so we don't need very high concentrations. Our concentrations can be in that range, 4 to 10 to 20%. So cement factories, which are about 95%, nat gas, 4 to 10, all that stuff can work. If you just map out the CO2 that's in highly insulated areas, high sun intensity in the US, there's more than enough CO2 next to areas where you can have water of sorts, call it saline aquifers and whatnot, to provide more fuel than the entire US, actually pretty close to what most of the world needs. So CO2 is very far from the limiting factor. If it were, then we'd have an issue. But the good thing is frankly, it's not. And there's a cyclical nature of this where the more energy we use, the more feedstock that we ultimately get.
Thank you. That was a very interesting presentation.
Thanks.
One of the things that you talked very early about disruptive technologies. And one question that I see is if you look at other approaches, say chemical approaches to making fuels, you see that there is a lot of work being done on, for instance, reverse water gas reactions. Butyrate chemistry could do very much the same sort of thing with biomass or some other carbon source. And given that material science has advanced so much in the last couple of years, people, of course, don't really like nuclear energy, but eventually someone may figure out that it's safer to be in a 9.0 earthquake and a 15-foot tsunami in a nuclear plant than in a building, which killed far more people. So you see the Chinese and the Koreans are doing all of this sort of high temperature chemistry. Isn't that a potential stumbling block to kind of because those things are continuous, and renewable energy, as much as people love it, always has the problem of not being a continuous process. Continuous processes are always more economic.
So I fully agree with your point, and we've actually tried to intentionally design all of our processes to be as close to continuous as possible, because I think it hits the nail on the head. In the opportunities where I have the ability to use chemistry, I tend to prefer chemistry over biology. It's sort of an arbitrary distinction. I just find that biology, because of its flexibility and specificity, and sort of, I hate to say it this way because it sounds wrong, but the knowingness by which you can say, I can get to a given product, makes for an interesting chassis by which you can at least start down that direction. I far from believe that biology will be the sole solution for the future. I think biology will be an important contributor in the fuel mix that we have at some point. I can't tell you if that's 25 years or 50 years or 100 years from now, but it has historically been in various forms. I believe that that's going to continue. Just the form factor, as it's changed in the past, will continue to change. We've invested pretty heavily in chemistry as well. I just didn't talk about that today. Midori, as an example, is a company that has pioneered an entirely new catalyst to free up sugars from biomass, and does so with incredible efficiency and renewable capacity. And it's been done in a way that I can't talk about, but that really is unprecedented in that space. And I think there's a lot of other opportunities. Novomer, for example, is also a catalyst approach to convert CO primarily, but also CO2 into common chemicals. Big advocate of that. I as much as fuels have historically been a zero-sum game and there's one winner, the future to me is not that.
I mean, I can see for high value added things, that might be a better approach, but for straight up fuels, the dimethyl ether, for instance, might even be a far better fuel than diesel. George Olov published in Journal of American Chemical Society, C1.
Yeah, well, so one of the rules I've learned about disruptive innovations, and I don't want to sound too much like Clay Christensen, is that you come in with a product that forces very little adaptation, and then you use that as a process to divert customers by changing the price, changing the spec as you go. And people use diesel today, they like diesel. A lot of the oil and gas companies have spent some time thinking about things like DME, but it's not a main part of our fuel stream. And since we're taking technology risk, I don't want to take market risk on top of that. I like, whenever I'm thinking about these things, to decouple my risks as much as possible. So part of the strategy, for example, we've taken with LS9 is not take, for example, the product that could have the single biggest impact, but take the product where we can have an impact, but get it to market. So then we can put the cost of scale and the cost of validation on someone else. That's what trips up most of these companies. And then use the chassis that's been developed and migrate into other areas.
Take one more question. I had a simple question. When you work in conversion of CO2, you presumably do that in the liquid phase, or do you use a separate CO2 carrying gas phase in the converter directly?
We've used.
The reason why I ask is, it looks like if you have two phases in a solar converter, it's basically unmanageable if you have a liquid and a gas phase, because you have to transfer both the gas and the liquid on an incline. But if you use work on dissolved CO2, then it appears to me the rate limiting step, because the solubility of CO2 in an aqueous phase is so small. So I just wonder, there is either a reactor problem or a rate limit problem in taking a gas into a solar converter.
Yeah, so there's a really tricky trade-off that one has to do there. We've done the gas-liquid thing. It works. It's chaotic inside there, and the modeling of it was a real tour de force by our team members who did that. It's controllable enough that we actually get some pretty good results, but we've actually moved away from that inclined system and to something that's a little less inclined. We do still use we still can, and in certain cases do use that gas liquid, because you get the benefit of movement and mixing. There's an additional cost because you're moving more, you're right. We've looked at, we've looked at dissolved, coming from more of a solid form in certain cases, and that works well as well. But it's a different flow paradigm because we put that in, the CO2 mixes very well, we don't have to worry about the interface, but then actually we have to provide energy for mixing. And the way we look at this is there's a lot of decisions we have to make on the deployment of on any given reactor. What the CO2 source is it local, is it far, what do you do with the oxygen, what do you do with the cells? And that our design, what we tried to do this is make it sort of modular from that component as well, that you make a set of trade-offs. And at the end of the day, all of these trade-offs will give you a certain set of economics. Those economics will be defined basically before you even build your plant. And the beauty is you can make an assessment and say, with all the choices I have to make to build a plant in location X, will I make money? And if you have to do certain things where you have to put in too much energy, or the heating load is going to be bad, or frankly, the cooling load, or whatever it happens to be, too much risk from insurance for hurricanes and tornadoes, don't build a plant there. So none of these are really just one or the other. We actually typically will validate both sides of any coin to see what works, what doesn't, what the trade-offs are, what the energy balance is, and how that comes together to make an equation.
Yeah, let's thank David again.